So today's talk is by the group of Professor Neelam, Sinha and her research scholars, and what promises to be a very interesting talk about the human brain. I guess their work has got a study has to do with the study of fMRI and MRI images and EEG-based time series studies. So the talk would be given by Professor Neelam Sinha along with three of her students. Gopika Gopan, she is doing her PhD along with Professor Neelam, and Deepanjali Bhattacharya. Right? So you're also doing your PhD along with, and Chandana is IM Tech student. She's working with them. So without further ado, let's take over. Let's get the speaker.
Hello everyone, thanks for showing up. I'm here at IIIT for the last 7 years, and the meat of my work has been on medical imaging and analysis. And most of what I've done is the sum total of what my students have done, so I thought it's only fair that we all should share the stage. So I'll only speak for those who are not here currently. I love this advertisement. It's a Siemens advertisement. There's a child looking at the skeleton of a dinosaur and wondering, is there a safer way to look inside our bodies? I think this really answers what medical imaging is all about. Are we really able to answer, can we safely study our bodies? So the kind of solutions that medical imaging provides, the different modalities, X-ray, ultrasound, MRI, no one guarantees 100% safety. It's only that the benefits outweigh the disadvantages, right? The medical image, the image there is a misnomer. It actually means it's a snapshot. It could be a 1D signal, it could be a 2D real image, it could be a 3D video, it could be a 4D signal, it could be in any number of dimensions. So the image there is just a misnomer. So what is a medical image? It's a window into the human body to see signs of disease and guide therapeutic procedures. What are the components that make it up? You should first know what is it that you're trying to study, the anatomy of interest. You build a sensor to ensure you capture the signal of interest. For example, if you're looking at EEG signals, your sensor will be the electrodes. You need to have signal processing incorporated. Why? Because what you're measuring is a signal. So you should have some guidelines on how to sample, what your sampling frequency should be, what the range of frequencies you're interested in. So that is determined by the signal processing aspect. So these together form the image or signal generation part. This is what Madhav typically does. Now, on the other hand, you have techniques of computer vision, you have machine learning algorithms. These together help you interpret what the image is saying, which part of the tumor is benign, which part is malignant. This kind of interpretation is what you're trying to achieve using computer vision and machine learning. So in any radiologist clinic, this part cannot be substituted. So this is fixed. It's only this that may or may not be there. Why is it? Because they still believe that they are the ultimate experts. We have a long way to go before we can reach their kind of interpretation. Now let's look at the imaging tools that we are speaking of. What are their desirable properties? I go to the doctor with a hairline fracture. I undergo some treatment, and after that, when he images me, no matter how small the fracture, how thin the fracture, his imaging device should be able to say if there is a difference or not, right? So this is what is called capturing fine details with fidelity. I've put fine here within quotations because each modality gives you a different kind of fineness. So how much fineness can you afford depends on several things. How good your equipment is, how much time you can allot to acquiring the signal, Several things go into that. But together they tell you what the spatial resolution of your image is. So if there's a brain with tumors, you want even the slightest tumor to be detected. So you really want your imaging modality to be fine. The next desirable aspect, as you can see, it's the plane of imaging. You want to have a modality that is giving you the flexibility in the plane of imaging. So these are the three standard planes: the coronal, the sagittal, and the axial. Now there could be certain parts of the anatomy that need somewhere in between. For example, while imaging the spine, it doesn't fit into any of these. You need to have an oblique plane of imaging. So your imaging modality, unless it allows that kind of imaging, how do you even image the details? So a good imaging modality will give you a very flexible plane of imaging. Another desirable property, can you guess? The safety. So you want the imaging modality to be as non-invasive as possible. X-rays are typically safe, but then pregnant women are not supposed to enter an X-ray room, right? So how much safety, how safe is safe, that's always a matter of debate. You need to have enough color levels to distinguish contrast. For example, if you're imaging a tumor, the tumor shows up even here, but then it's not very prominent. So you need to have a modality that will emphasize the tumor, emphasize what you're looking for. Another desirable property is your modality should be fast enough so that the physiological dynamics are captured. So this is called what is temporal resolution. For example, if you want a video of the beating heart, you have to ensure you're fast enough to capture the entire physiology there. Now, when we image not just the structure, you could image even the functioning. So this is the functioning of a normal brain, a mildly impaired brain, a severely impaired brain. So here what you're capturing is not the anatomical detail, but the metabolic detail. Functional MR imaging, you put a subject in the MR scanner and ask him to perform a task. It could be an auditory task where he's listening to maybe some music. It could be a visual task where he's looking at a picture. It could be a motor task where he's moving an arm, or he could simply be taking rest. So the person in the scanner is performing certain well-defined tasks. And what is the scanner doing? At every timestamp, it's capturing a volume of images. It's as if the entire brain is sliced like loaves of bread, slices of bread that you see in a loaf. So one volume would correspond to the entire brain volume taken at timestamp T1, so on for T2 and so on to Tn. So you end up having a 4D signal where at each timestamp you have a volume of images. You do some kind of analysis on this to be able to tell something about the task that the person was performing. So here what you're capturing is functioning of the brain. You also have something more sophisticated, looks so beautiful. So what's inside the soft tissues are the neuronal tracts. Is it possible to image them? It is. So this is called diffusion weighted imaging. And here you're capturing microstructural details of the neuronal tracts. When would this be useful? In certain ailments, there could be a disruption in the tracts. So if you want to know if continuity is lost because of that, they typically do this kind of imaging. Now with all this, is mind reading possible? You put together the different modalities that you have. You have functional MR imaging, you have MEG, you have EEG. It is possible to know what the person is thinking, but not the subtle kind of imaging. Not like the poets and philosophers we have here, but very simple dichotomies. Is he thinking left or right? Is he thinking of a noun or a verb? Is he thinking of an ambiguous sentence, unambiguous sentence? So such simple dichotomies can be differentiated with the modalities that we have.
There was a webinar I saw from some lecture.
Yeah.
He was even talking about even objects having very kind of precise signatures.
Yes.
Which are very consistent across
Yes, yes, yes.
Wouldn't that help you go even beyond this?
No, but I'm telling not the subtle kind of thinking, Mane, not like Shakespeare or Kalidas. Yes, 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 yes. Thinking of a noun in English versus thinking of a noun in Chinese, even this can be differentiated. Yes, yes, yes. That's why they say that it's very much advisable that the child learns multiple languages, right? So let's see what are some of the challenges we face in working with these images as opposed to working with natural images. There's a lot of diversity in image quality. Why does this happen? It depends on the kind of sensor we are using. It depends on the kind of image that we reconstruct. Now, typically in these modalities, you don't get data in the image domain. You get it in some other domain which needs to be transformed back to get your image. So a lot depends on how good your sensor was and how good your image reconstruction algorithm was. With the same data, I can end up with one image and you can end up with another. So there's a difference in consistency. And these are typically very, very noisy. So communication people would be very scared of such SNR levels. 10%. So it's noise of the level 100 and my signal is 110. So I just have that margin to differentiate my signal from noise. Very hard to straightforward apply any of the signal processing techniques here. To get a good image, even when we click on our mobile cameras, you want the person to be still. Now, what if he has to undergo scanning for several minutes? Can you expect him to be sufficiently still? It's not possible. And in the process of trying to acquire the data, there could be some coupling of the fields within the body with the sensor itself. So the uncertainty principle comes into play here. And it's very hard to find uniform or regular sized image patches for measurements like volume, dimensions, texture, the way you would easily find in natural images. It's just not possible here. And it's hard to have a standard image atlas across population, across races, across ages. And for the same person, same location, there's a lot of variation in data across samples, across time. So generalization becomes very difficult. There's no benchmark. Yeah. 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 No, it does, but with a pinch of salt. So even now, for our kind of brain measurements, people use Caucasian atlases, which is just not good. So there's a lot of work going on in trying to create an atlas for Indian population. And even there, for infants, it's different, for toddlers, for teenagers. Now, apart from those challenges, there are other challenges that we face. Data availability. We need reliable studies. We need loads of well-acquired data, which needs a lot of clinical precision. And you need appropriate labeling, which is where our medical collaborators come into play. And we are so glad that there are lots of publicly available data repositories. That is what gives us the initial ammunition to work with. And we are very grateful to our collaborators at NIMHANS. They really help us out. And even before data availability, the very first thing that they help us with is they give us a problem of relevance. They tell you what is important, what is the clinical world looking at. So that is the first thing that we are very grateful to them for. And after we have done our work, we need the results to be validated by them. Here I want to pause for a moment. I want to acknowledge the support that establishing EHRC has provided. And with MinRO coming up and the emphasis on healthcare, I feel so proud that I chose this domain to work on. So we are really looking for opportunities to grow and showcase our work in several different ways. Now the outline of the talk. So most of the talk will be on brain studies using different variations of MRI. First the functional MRI, the diffusion-weighted MRI, structural MRI, Eeg time series, and then the last bit will be on analysis of surgical videos. Now, we saw this in the introduction. Functional MR is when you are trying to image functioning of the brain. You have a subject in the scanner who is asked to do a well-defined task. And I'm repeatedly emphasizing on well-defined. One of these typically, and at every time stamp, you end up with a volume of images. Now, how does FMR work? There's no contrast injected. How is it that you get to know how the brain is functioning by capturing these images? So what is important there is what is called BOLD contrast. BOLD stands for blood oxygen level dependent contrast. We'll see what it means. When the person is at rest, the blood flowing through his neuron has a good mix of what is called oxygenated blood and deoxygenated blood. It's almost at equilibrium. So when you focus on any neuron there, you don't see any activity happening because he's at rest. Now the moment you ask him to do something, there's a spike in neural activity which makes more blood flow towards that neuron if it is involved in that activity, which means there's a rise in the level of oxyhemoglobin, which leads to a rise in the MR signal. So it is this sequence of events which fMRI captures. So now the same picture, now you see there's more of oxygenated hemoglobin moving through it. So this will result in a contrast here. So if this neuron is occupied in this activity, it will show up as bright. So these task-based cortical mappings, these are very well known. So how do you utilize BOLD signal? You know for a specific task. If it's an auditory task, there's a specific region of the brain that is involved. If it's a visual task, there's yet another part of the brain that is involved. So these mappings are very well known, very well understood. So if you're listening to a song, this part of your brain is involved. So now if you're really listening to me, it's this part of your brain which is active. You sing, there's another network that is functioning. So depending on the activity that you're doing, which part of the brain gets activated differs. So all that analysis is to obtain what is called the activation map. So you analyze the time series of images. When you pick up a voxel that has been active in the activity, when it is active, you will see the level rise. When it is at rest, you will see that it's not, right? This is the baseline level and this is the activity level. Now this is a very exaggerated, filtered out time series that you are seeing. In reality, it's not this clear, which is why the entire challenge comes. So at the end of all this, what you obtain is called the activation map. This will tell you that these parts of the brain were active during the activity. So this kind of analysis is being done since 90s. So Friston was the first to pioneer it. So what he did was he looked at each voxel independently and analyzed each time series separately. And then as times changed, Tom Michel, he's been a very big pioneer here. He spoke of looking at the entire volume of brain as a single pattern. So now if you have say 5 lakh voxels in the brain, you have a pattern made of 5 lakh features. Now this is any machine learning student's nightmare. So you look at patterns with say 20 features, 50 features, 100 features. Nobody works with patterns which have 5 lakh features. So it turns the entire machine learning theory on its head. So he has done a lot of experiments, Tom Michel, and he's also put up a data repository online that we too have used. And slowly, when the field changed, they came up with what is known as finding region of activation by Searchlight. So it's been around since 2006. So what they do here is they look at a small region of the entire volume within which they analyze. So you don't look at each voxel separately, you look at a bunch of voxels together. So what was the advantage? They said this analysis produces maps by measuring information in small spherical subsets centered on every voxel. So now you have some context also. So you know that no region of the brain could be acting isolated. So you look at the neighbor's information and then decide on what the central voxel is doing. So the map's value for each voxel is derived from the information present in the entire neighborhood and not individually by the voxel itself. But this approach has several limitations. One will never know what the appropriate size of the window is or even the shape. It needs exhaustive search across the entire volume. It's an overkill. And if you have signals from large dominance regions, you could end up ignoring subtle signals which are coming from elsewhere. So there are several limitations of this kind of approach. So my first MS students worked on this problem, and some of the questions that we tried to answer then, way back in 2012, what if the anatomical mapping of the given task is unknown beforehand? So if you don't know, you don't even know where to look for it. It's a very vast search area. And can you rank brain regions with respect to relevance? Can you say this is the most important part, this is the second most important, third most important, and so on? And all along, people had been looking for region of activation. So we were trying to say, if the person is performing two different activities, not at the same time, he does one activity once and later does another activity, can I localize the region of discrimination between two activities? So these were the questions we tried to answer. We came up with a very naive solution. Still, I thought I should present it here. That was the first work we did. So what we proposed was differential weighting and iterative classification. You look at the entire brain volume. So the key thing is not to ignore any part, because you never know which region may be important, even if it is subtle. So you divide the brain volume into 4 quarters. It could have been 8, it could have been 2. We just happened to choose 4 quarters. So now the entire volume is divided into 4 quarters. You run a classification algorithm, any classification algorithm of your choice, with equal weightage to all of the voxels separately in each of the divisions. Now, if there is one part which is very prominent in the activity, I expect that part to perform very well. Now, fine-tune the weightage of the voxels based on classification performance. So, if this quarter works well, I will assign more weightage to those voxels. Some part does the worst, I'll ensure that the weightage given to those voxels is the least. Yeah. So I know that the person has done 2 different tasks, and I have the ground truth. I have some ground truth. So we will fine-tune weightage of voxels based on the classification performance, choose the best performing quarter, and further repeat the same exercise. Now fine-tune the weights of the voxels of the best performing one. So this way you can reach those voxels that are the most relevant. Now if you want the second best, you leave out this and then repeat it for the remaining. So in essence, this is what happens. As iteration progresses, your search space gets smaller and smaller. So the features we used, now when you look at voxels that are involved in task 1, what are the components? The baseline signal itself, the activity related signal, and noise. And now these experiments are always conducted with activity happening in one phase followed by a spurt of rest, and then again there's activity followed by rest. So when you look at this, so you have voxels, you know those timestamps that are taken from rest, you know timestamps that are taken from task 1, you know timestamps that are taken from task 2. So you look at where the signal is different from task 1 and rest. That will give you an idea of voxels that are related to task 1. You do the same thing for task 2 versus rest. You get to know voxels that were participating in task 2. So we tried this experiment on 2 publicly available data sets. So Star Plus data set by Tom Michel. The tasks we looked at were the negation task. That is, the subject is shown a picture and he's asked to read a sentence that may or may not represent that picture. So if the sentence does not match with the picture that he was presented, it's called a negation task. And if the sentence correctly describes the picture, it's called an affirmation task. Another publicly available data set that we used is the PROBIT data set. So this has 3 tasks: viewing pleasant pictures, unpleasant ones, and neutral ones. So in both the cases, we try to discriminate between here we try to discriminate between these 2, here we try to discriminate between these 3. And these are our sample results. Without attaching any weightage to the voxels, that's the baseline classification. Once you fine tune weightages to the voxels, what you get are relatively better numbers. And our inferences from here, that the relevance of the voxel is reflected in the weightage that gets assigned. It's applicable to tasks about which we have no prior anatomical information. And this can help us localize regions that lead to discrimination between tasks. Now we'll move on to the next work. This is on diffusion weighted MRI. It's a more recent work carried out by a PhD student here, Rajika Raja. So this is called assessment of tissue heterogeneity using diffusion MRI for grading gliomas. And Nimhan's collaborators have helped us in a very big way here. Let's see what is diffusion MRI. Now diffusion itself is movement of molecules from region of high concentration to a region of low concentration. And what is the objective? The objective is to image microstructures in brain by quantifying this movement. So if some region has very high diffusion, you will see that it will decay very quickly. If some region has very low diffusion, the signal strength decays not very quickly. So different tissues have different properties of diffusion, which is what we characterize here. So the model used here is an oriented ellipse. It fits in well because most of the neurons, dendrites are all oriented ellipses. So most of our brain tissues are all oriented structures. So there is restricted or direction specific diffusion. Because the ellipse forms a barrier, it cannot diffuse in all possible directions. Diffusion has to be oriented, and diffusion can be exploited to determine presence or loss or change in structures. So you use the information of diffusion to compute what are called the parametric maps. The parametric maps could be diffusivity map, anisotropy map, several other maps. So in neuroimaging, it's typically used to study neuropsychiatric conditions related to aging. So if this is what a structural MRI looks like, as you know, there are 3 very distinct regions in our brain: gray matter, white matter, and CSF. White shows up as white, gray matter shows up as gray, CSF is in between. Now, when you look at a parametric map that you obtain using diffusion imaging, for example, this is the mean diffusivity map. It looks very different from the structural one. It's not capturing structural details the way we see it here. What it captures is something else. It's capturing diffusivity. So if a region is very bright, it means diffusion there is very fast. And if a region is very dark, it means diffusion there is very slow. Similarly, this is the fractional anisotropy map. Isotropy is being spherical. Anisotropy is not being spherical. So it tells you how anisotropic a particular point is. So if there is a region which is very dark, it means it is highly isotropic. If there is a region which is very bright, it means it is highly anisotropic. So these differences are what are captured in diffusion weighted imaging. Now we use this for glioma grading, as we said earlier. Let's try to answer what is glioma. Glioma, as you can see, is a brain tumor. World Health Organization has defined grades 2, 3 and 4. The number of the grade indicates severity of the tumor, and it's important to determine the grade to decide the course of treatment. Higher grades of tumor manifest higher degrees of heterogeneity. By heterogeneity, we mean being uneven. You'll see that. So the objective is to provide non-invasive and assistive analysis to the radiologist. Now across grades, now grade 2 is fairly even, whatever you may see here. Grade 3 is not so much, but grade 4 is highly uneven. So the objective of the work was to see if we can capture this information through images. So you have the entire volume of the brain in front of you, and the tumor may be spread across, let's say, 3 to 4 slices. So in each of the slices that it shows up, in one slice it may look small, the very next slice it may look a little large, and after that if it's diminishing, the next slice will be smaller and so on. So you have to integrate information across slices. So you need to capture 3D spatial features across X, Y, Z. Now texture quantification is not a challenge at all on natural images. There are several tools available for it, the Gabor filter banks, wavelet transforms. All these are possible because you can have a well-defined patch within which you can capture texture. You also have measures like entropy, busyness. Now these are statistical measures. They don't capture the spatial layout of your data points. It's as if they were all 1D points spread out and you're capturing those. So we know this is not good enough and this cannot be done. So we needed to come up with a technique that could quantify texture across an irregular shaped 3D volume irrespective of the size. The tumor may be small, may be big, you never know. So we proposed what we call the volume heterogeneity index. So we based it on cross correlation. CC stands for cross correlation here. So this was the observation that we had. If I look at the cross correlation between two even pulses, then the resulting thing is a very smooth triangle. Now if I make one of them jagged, the resulting triangle is slightly jagged. Now if I make both of them jagged, the cross correlation is highly jagged. So we thought if this can capture the essence of unevenness, we'll be through. So from this jagged plot, what we did was we computed slopes at different points and looked at the variation in it. So we looked at the variance of the first derivative of the cross correlation. It's a very simple idea. So, okay. We computed parametric maps, and on the parametric maps, we computed these cross-correlation plots. And we did this in all the three planes, axial, sagittal, coronal, just to make sure that all three are in sync. This is information of the kind of subjects, the age range, the male-female ratio. This is what we could get from our collaborators, so we couldn't do more than this. So we made sure there was enough data to estimate diffusion tensors well enough. And the ROI selection, the region of interest, the tumor selection, that was done by the radiologists themselves. So a typical case looked like this across all the three planes, axial, coronal, sagittal. The tumor was marked out. So we had to work on these regions. And when you look at the cross-correlation plots, these fall exactly in line with what we had envisaged. A even tumor will give this kind of cross-correlation plots. Slightly uneven will result in a jagged plot, and highly heterogeneous will lead to very, very jagged plots. So the inference is that volume heterogeneity index is indeed effective in distinguishing tumor grades. We'll move on to the next work, structural MRI. Yeah. There's a lot of pre-processing. So diffusion values that are plausible for a human subject. That itself, even when you compute it, there are so many numerical problems you encounter. Yeah. Our collaborators were very happy with this work. I can tell you so much. By the our radiologist. Yeah. In some of them, actually, there are the cut tissue cross sections. Yes. They actually, yes. The labels are what are hardest to find. Now, we'll take a small detour before I hand it over to Debanjali. We'll just move a little away from brain studies. I won't take more than 3-4 minutes for this. We've done some bit of work on fundal image analysis. Fundal refers to the eye. There are like unrelated works. We need to put them together and make it concrete. So one of them is by one of our Sansan students, Kishore. I don't know if you can see the details well enough, but this is the picture of a normal eye. And here, this is the picture of an eye that is affected by what is called diabetic retinopathy. So what distinguishes diabetic retinopathy eye from a normal eye is the formation of these. These are called hard exudates. So he worked on, first of all, you should be able to say if the eye is normal or affected by diabetic retinopathy. And if it is affected by diabetic retinopathy, localize where those exudates are. So he used deep learning for it. Most people in medical fraternity, they frown upon deep learning because they are not able to interpret results. So what he used was AlexNet. He used the good old Kaggle data set of 7,000 plus images. And he extended the idea of what is called CNN fixation. Now what is fixation? Now if I give this image to an image classification network and it returns the label rabbit, it is saying so because these pixels belong to a rabbit. So fixation refers to pointing out those locations in the image that constitute the rabbit. So given this image, if I get the label rabbit, it will tell me that these are the pixels which are responsible for that label. So a heat map on this will tell you that this is the region in the image that led to that label. So this is the idea we used even here. That is, if this image gets classified as a diabetic retinopathy affected image, it is happening so because of these. So this is the idea that he used. And these are our sample results. I don't know if you can see clearly enough. You see exudates all over, and he indeed got exudates here. Yet another sample result. So the best way of quantifying performance in our kind of work is to look at the ROC curves, the region operating characteristics curve, and the area under the curve he got was 98%, which is actually very good. So yet another work on the eye images was done by our PhD student. So if this is the eye image given to you, can you come up with an algorithm that will give me only the blood vessel network? Anyone who's done any computer vision knows how hard it is. There's non-uniformity in the structure. There's loss of continuity in several places. There are illumination differences. So there's a lot challenging here. So he again used a deep learning framework. He used a publicly available data set for it. And he processed the images patchwise. That is, he cut it into sub-images, and in each sub-image, he located where the blood vessels, and then stitched them all together to get his answer. So the output is a binary image, which should show 0 if it is not a vessel. It should show 1 if it is a blood vessel. And these are his sample results. And as I said earlier, the area under the curve of the ROC curve is 0.89, which is very good for our kind of. I'll hand it over to Debanjali now.
Good afternoon, everyone. So, my research topic is quantitative image analysis on neurological disorder. So, what is neurological disorder? As we know, the brain, spinal cord and nerves makes the nervous system, and together they control the working of the body. So, when something goes wrong with one part of nervous system, we can have problem with speaking, learning, memory, etc. Now, there are more than 600 neurological diseases. Some common types include stroke, meningitis, epilepsy, brain tumor. Now, in my research currently focuses on two types of neurological disorder. One is neurodegenerative Parkinsonian diseases and brain tumor. For the today's presentation, I will briefly discuss the topic on what I have done on neurodegenerative Parkinsonian disorder, and we have done quantification of changes in corpus callosum for these Parkinsonian diseases using structural MRI. So, what is neurodegeneration? So, neurons, we know neurons are the nerve cells, and they have the ability to gather and transmit signals, and there are more than 100 billion neurons and many more neuroglia to protect that neuron. So, neurodegeneration means the progressive loss of structure or functions of neuron, or often the death of neurons. Now, what are the causes of this neurodegeneration? The first one is aggregation of misfolded proteins. So, protein folding, that is the physical process by which protein chain acquires a 3D structure to perform the biological function, and if it fails to fold in a desired structure, it may lose its normal operation. Then accumulation of intracellular toxic proteins due to mutation. So, mutation causes changes in DNA sequences, and DNA contains the information to make protein. So, any changes in DNA sequence may, for that this toxic protein may accumulate, and that may alter the biological function, cell death in any form. Now, it is seen that aging, that is the greatest risk factor for neurodegenerative diseases. It is seen that most of the neurodegenerative diseases are late onset. That is, there are some factors that changes with aging. So, the process of neurodegeneration, it is not well understood, and most of the cases there is no cure. So, the challenge is to design new biomarkers that can track disease progression and staging, to investigate new factors that may contribute the diseases, and so on. There are many quantitative studies that aim to detect the changes in whole brain structure, several cortical and subcortical brain structure. In my research work, instead of whole brain analysis, my research work concentrated on a particular brain structure which is called corpus callosum. So, what is corpus callosum? So, that is shown in this figure. So corpus callosum, that is the largest white matter structure that connects to cerebral hemisphere and enables all kinds of interhemispheric communication. And why it is called white? Because it is the tissue with high myelin content that covers the axons. And this white matter is a tissue through which the messages passes between different areas of gray matter within central nervous system. So any changes in white matter are associated with neurodegenerative diseases. And as corpus callosum is the largest white matter structure in brain, so any subtle changes in corpus callosum may correlate with cognitive and behavioral deficits for these neurological diseases. So for my research work, I have used sagittal images. So this is the corpus callosum. So why sagittal images? Because in this plane we can visualize the whole corpus callosum structure anterior to posterior, which is not visible properly for axial plane or coronal plane. The data set that is collected from NIMHANS, and the data set included patient groups having Parkinson's disease, having multiple system atrophy and progressive supranuclear palsy. So these multiple MSA and Psp, these two are the atypical Parkinsonism, which means they have some common symptoms with typical Parkinson diseases like rigid and stiff muscles, loss of balance and coordination. But these diseases have some additional symptoms. So that is why it is called atypical Parkinsonian disorder that can distinguish them from simple Parkinson disease. So total 64 adults are recruited, 16 in each group. So the T1 weighted is at the data set. So this is the brain for a healthy control, one subject. These are the MR images of Parkinsonian disease, of PSP, of MSA. So as you can see, for healthy control group, the corpus callosum structure, it is much thicker, whereas for other disease group, the corpus callosum structure, it is thinner. So the goal is how to quantify this reduction, how to quantify this degeneration. So the aim is quantification of structural changes in CC. So we have quantified the changes in CC structural features like thickness, area and length. So the dimension considered to quantify the CC structural changes is thickness, area and length, and C8 software is used for the measurement. And statistical analysis is performed where we have checked the hypothesis that there is the regional anatomical structural damage in corpus callosum in disease group compared to healthy control, and which is followed by SVM classification. So C8 toolbox is used for paracelating the mid-sagittal corpus callosum into 100 equal segments, measuring the thickness and each segment. So by a series of 99 radial lines, it divides the entire corpus callosum into 100 segments, measuring the thickness. And then this thickness profile is plotted. So the blue dotted line shows the thickness profile of a healthy control, whereas this red, black and green line shows the thickness profile of the disease group. So if you can see, the thickness is much reduced here compared to the healthy control. So how to quantify this? Similarly, we have measured the area within geometrically 5-partition scheme of Wittelson and Hoffer. So Wittelson and Hoffer's classification, how they differ. So according to Wittelson's classification, say for an example, the first region 1 consists of 1/3 of the total corpus callosum. So what it means? It means that the fibers which are passing through the corpus callosum are connected to the prefrontal, premotor and supplementary motor area of our brain. On the other hand, Hoffer classification, the first region 1, it consists of 1/6 of the total corpus callosum. That means the fibers which are passing through the corpus callosum is connected only to the prefrontal region. So these two are the well-known classification scheme that is used in our study. So the area profile for the subject, for the static groups, that those are plotted. The length is also plotted, but for length it is seen that only MSA, the length of MSA and healthy control, those are statistically significant, but length of Parkinson, PG, PSP, those are statistically not significant, but area profile, that it shows significant result. So the HVM classification, I have presented only based on thickness, whatever we have got, because this is the highest classification accuracy that we have obtained. Okay, thank you. I am handing over to Gopi.
Good afternoon. I'll be giving an overview of the EEG based research at IIIT. So before starting that, what is an EEG signal? So EEG signals are electrical signals which are generated in the brain due to firing of a neuron. So this is a sample of an EEG signal. As you can see, there is no definite pattern as in the case of the heart signal, which is ECG. So it seems totally random. So in literature, we have found that 0 to 60 hertz frequency is the one which contains the most useful information to characterize whether there is any disorder or which state of the mind the person is. This 0 to 60 hertz frequency can be further divided into subbands, that is beta band, which is from 14 to 30 hertz, which is associated when a person is in a highly active physical mode or a highly active mental mode. The second is the alpha, when the person is awake but in a pretty relaxed mode. The third is delta, which is below 4 hertz, it's when a person is in deep sleep. Then we have theta, which is 4 to 8 hertz, which is usually associated when a person is in a drowsy state. And finally, we have a gamma, which is 30 hertz and above, and it's associated with heightened perception. Usually, it's said that when we have a lower amount of gamma, the person is more prone to learning disorders. So, this is a sample of how a normal EEG varies with the disorder EEG. Here I have chosen seizure, that is the fits, when a person experiences fits, that is known as seizure. So here you can actually clearly see how different the EEG, normal EEG is from a seizure EEG. Now there are two types of seizure. One is partial seizure, where only certain part of the brain abnormally fires. And the other one is generalized seizure, where all part of the brain abnormally fires. So this is easily captured in the EEG, as only some of the channels have got abnormal EEG. Here all the channels have abnormal EEG. So why EEG? We have seen that there is MRI, fMRI, diffusion MRI. So why EEG is, first thing, it also gives an insight into the functioning of the brain, and also for each type of neurological disorder or a mental state, there is a specific characteristics. The most attractive feature of EEG is it's non-invasive, inexpensive, pretty painless, easy to do procedure, and recently there is commercially available mobile EEG headsets such as NeuroSky, Mindwave, eMotive. So this makes it easy to get our own data. So the data that we have used so far in our research is first is from online repository, that is UCI machine learning repository, where we have the data of a long-time alcoholic patient with a control patient. The second is Bonn University repository, that is ICTEL. ICTEL is same as seizure, the data when a person is having a seizure, versus INTER-ICTEL, a person who is known to have a history of seizure, but at the time of taking EEG there is no seizure, and a normal person where a person doesn't have any history of seizure. Then we have Physionet repository, where we have the eyes open versus eyes closed. This is not the process of eyes opening and eyes closed, this is the baseline. Then we have sleep stages, then deep database, where we have emotion classes, arousal, dominant, valence. Arousal usually measures the intensity of the emotion, for instance anger and rage. The rage has got a higher arousal state. Then we have dominant, it's like the dominant nature of the emotion. In case of anger and fear, anger has a dominant nature. Then we have balance, that is usually associated with the goodness or badness. For example, the joy is a positive valence, anger and fear is a negative valence. In addition to this, we have in-house data collection, where we have collected the data according to the principles of Declaration of Helsinki, and we have used single channel EEG NeuroSky Mindwave device, and the experiments were formulated based on the information that can be available through a single channel. Overall, the EEG based research can be divided into two types. The first, we have data analysis. Here I just give an overview of statistical features and source distribution. Then we have data model. I'll just briefly say what neural mass model is. So initially we have data analysis, two types, statistical analysis and source distribution. These are mainly used in classification of a given EEG into specific classes. So in statistical analysis, initially we have a raw data. So this is how a raw data is a single channel followed by feature extraction. It can be statistical features, wavelet decomposition followed by statistical features. It can be chaos analysis also. Then we have classifier, SVM, naive Bayes, all those. And finally, class 1 to class K, it can be ictal versus normal, eyes open, eyes closed. This is a generic overview. Based on this, we have done some experiments on earlier said repository. So the first one is alcoholic subject. So here we can, this is the control subject and this is alcoholic subject. Here you can clearly see there is some subtle differences between the two types of EEG. We have used here hybrid features, that is we have combined the raw measurements along with some statistical features, and we have been able to obtain accuracy of 88%. Here the statistical features were mainly from theta band. The next one was ictal versus intraictal versus normal. As I told earlier, ictal is when a person is experiencing fits. So at that time you can see there are specific patterns being formed. So here we have spike wave, which is usual characteristics of a seizure. Intraictal is the time between two seizures of a particular person. That is, a person is at present not having any experience of seizure, but he has a history of having seizure. In such a case, there can be isolated spikes being located. And finally, we have the normal EEG. So here again, it was found that statistical features, especially the entropy, was the most effective one in distinguishing the three types of classes. And using a feature ranking, relief F, it was also seen that entropy was best suited. So far we have seen that the different classes we can visually see there are certain differences. So right now we have come to two classes, eyes open versus eyes closed. These are the baseline. So as you can see, we don't visually see much of a difference between the both. That is, visually it is not seen. So we need to see whether statistically we can find any kind of differences or any kind of distinguishing between these two classes. So here we did statistical features, and it was found that the skewness and kurtosis of the data were much more effective in distinguishing the two classes, and in the feature ranking, skewness scored the highest. Apart from this, we have also done some statistical analysis on our in-house collected data. For example, attention, relaxation, creativity. These are single channel data, and here also statistical features proved to be much more efficient. So these are just highlights on some of the statistical analysis. Now moving on to source distribution. We need to know whether the source of the data, the distribution of different classes have any kind of difference. So one of the examples that I have chosen to illustrate this is eyes open and eyes closed, because visually itself we don't see any difference in that. So the method is, we take the data, we do feature extraction, that is statistical moment. We calculate the moment generating function, which is actually given as a feature to classify it. So here again, we find that the MGF is effective in distinguishing between this, and a sample of the moment generating function for the two classes are as follows. As it can be seen from the figure, they are quite different. So that was about the EEG data analysis. Now EEG models. Why we require EEG models? We need to know what exactly is the process that actually lead to certain type of EEG. So for that, there are two types of EEG models. The first one is a single neuron. You start modeling from a single neuron to a number of tissue to the entire brain. So it's like computationally pretty expensive. The second one is macroscopic model, where we try to analyze a group of a neuron, how it behaves. So we'll be focusing on macroscopic level, in particular neural mass model. So what a neural mass model it models is a cortical column. What is a cortical column? Over here in the brain, if we take a small part of the cortex, we can see histologically there are 6 different layers, each having different types of neurons. So this is known as a cortical column. And neural mass model actually models this cortical column. There are different types of neuron present here. We have got interneuron, which is usually connecting the neurons of different layers. Then we have sensory neuron, which is associated with the senses, motor neuron, which is associated when we move the legs or hands. Then we have pyramidal cells, whose output is what we achieve as the EEG on the scalp. So, neural mass model is based on two main transformations. For instance, this is a sample of a neuron. We have got dendrite, nucleus, cell body, the myelin sheath, which enables the action potential to jump over and move fast, and axon terminal followed by a synapse. So, the neuron before the synapse over here, it generates some neurotransmitters. These neurotransmitters can be excitatory or inhibitory. So that means it can increase the amount of the value or decrease the amount of potential value when it reaches the dendrite. So when it reaches the dendrite, it creates a potential which is known as postsynaptic potential. So this excitatory and inhibitory PSP together they sums up, and if that value exceeds a certain limit, then we have action potential being generated at the hillock over here. That is where the potential to rate transformation occurs. So this potential to rate transformation is generally nonlinear in function, and we usually take a sigmoidal function to represent it. So this is the equation where we have E0 as the maximum firing rate. Then we have V0, which is the average potential that is for which the half of the population fires, and R decides on the steepness of the sigmoidal parameter. Next we have Psp transformation. It's a linear in function, and this is the equation for Psp transformation. Here the peak and the time at which we have the peak is controlled by this capital Q, which is synaptic gain, and small q, which is time constant. So neural mass model basically works as a convolution of these two transformations. That is, we have got H here, which is the Psp transformation, S here, which is the potential to rate transformation, and Q is the synaptic gain. So if we do an Laplace transform analysis, we will end up with a differential equation of this form, which can be further analyzed. The next, I will just show simple two common types of neural mass model that is used at present. So we have Janssen and Red model, which is 1995 model. We have got three sets of neurons, that is pyramidal cells, out of which we are obtaining the EEG. We have got inhibitory interneurons, which inhibit the pyramidal cells, and excitatory interneurons. So further detailed analysis is shown here, where this shows the sigmoidal function, that is potential to rate function. We have over here the PSP function. All the C's represent the amount of synaptic connection between each of these populations of neurons. So this is one. The next one is Bendling Model, which is a 2002 model. So here he has done two types of inhibitory interneurons. The reason for this is he found that the neurotransmitter, the inhibitory neurotransmitter, is actually present in the dendrite as well as the soma of the neuron. So there are two different types of inhibitory neurotransmitter being present. So whichever neurotransmitter inhibitory present near the soma, it has got a faster activation and a faster decay rate, while those that is near the dendrite has got a slower activation and slower decay rate. That is why there are two types of interneuron present here. So these are the basic two models, and some of the active area of research with respect to neural mass model is, first thing is estimation of different states. States in this amounts to the average membrane potential. Then fitting a real EEG into the model, we have simulating the characteristics of EEG with respect to neurological disorder, then understanding the different connectivity of the brain, and so on. So this basically concludes the overview of what exactly is the EEG research going on here. Now moving on to Champana.
My work is broadly in surgical video analytics. So given a video of a neurosurgery performed by a neurosurgeon, can we inference something out of this? Can we analyze the video to give some feedback to the surgeons regarding how well they have performed the surgery? So this majorly is associated with visual cues that we obtain from the videos. So given neurosurgical videos, there are a number of tools used in this particular video. And what we're trying to analyze in these videos is primarily tool usages. So patterns within tools, and if they can say something about the surgery. So given every frame in the video, can we say that there are two tools used in this frame, and this is tool A, this is tool B, and kind of segment them out of the frame. So in the data set that we have obtained, it was annotated by doctors at NIMHANS. These are neurosurgical videos of patients from NIMHANS Hospital. So there are 5 tools used in all the surgeries. And the suction tube is used to suck blood out of the tissues. Syringe needles are used to make really small incisions. Dissecting forceps for making small incisions again. Bipolar forceps for grasping tissues, and scissors is a tool which is used very less. And with scissors, you typically end up making more incisions. So doctors typically avoid using the scissors tool. We pose 2 tasks with respect to this particular problem statement. The first is multi-level classification of tools from surgical videos, which helps us in evaluating surgeries. So in a sense, we're trying to create a framework which can be used to evaluate surgeries and give doctors feedback on how they have performed the surgery. And the second task that we have taken is tool segmentation, which goes hand in hand. So for tool classification, the primary challenges that we have faced are very related to data. Multiple times there are tools offloading each other, because of which the classifier may not learn the visual features well. Then there's an amount of variable zoom, because doctors might zoom in the video to locate parts which are more relevant to them during the video. Then there is always a blur because they hastily move around tools. Then there's reflection because they're metallic in nature and they might reflect the tissue as well. Then there's an immense data skew. Scissors is a tool which is rarely used, which they don't prefer to use, and only 3% of the videos had scissors. So this kind of made the training hard. In addition to this, the tools that the doctors actually use are millimeter in thickness. So what they typically do is they make an incision in the body, and this whole thing is displayed on a monitor. And looking at that, they perform the surgery. So it's important to see what tool they're holding in which hands in case they don't want to make any mistake. So there's also a very huge domain shift, because most of the classifiers are trained for general images in which the lighting conditions are very different. For example, sceneries will have a very different set of colors when compared to medical images. So the first aspect is of domain shift. So to account for that, the approach was to take a state-of-the-art classifiers and train it for an NIMHANS dataset. But before that, we trained it on neurosurgical tools dataset, which was made publicly available by a medical university, and tried to train the classifier to suit for the contrast in colors in neurosurgical videos. So this helped increase the accuracy by around 4%. Then we haven't done any amount of data cleaning, because in real life the data will come with a good amount of blur and occlusion and so on. So we wanted the classifier to be robust enough. For that, we have also done data augmentation to increase the amount of data that we have. And as I've mentioned, scissors was occurring in very few amount of frames. So we did. Yeah. Yes. It might seem like a very silly thing, but this actually happens in reality. So, yeah, that was the first thing that the doctor said. Then the other thing was feedback, and also it has extensions towards robotic surgeries. So it detects tools and areas where the tool is present, and there are also problems like depth estimation, so that a robot can automatically perform the surgery. So. So we obtained good results. We got an F1 score of 0.85 for classification, which is pretty good considering the amount of blur which is there in the images. So typically we want to obtain maps like this which talk about the timestamp of the surgery and the various tools used over here. So you see that sometimes between using tools there is a switch time between tools, which is not a very good indication because the doctor is using too much time to switch between tools, and you typically don't want the tissue to be exposed to the outside atmosphere for a long time. So all these things like tool switch time can be quantified, and also things like how long the scissors tool is being used. So we have showed the output to the doctors, and once we can quantify these, we can quantitatively also express how the surgery was performed. But by looking at this, you'll be able to say that this is not a very good surgery because they've used too much time to switch between tools, and there's also a good amount of usage of scissors.
I'm also wondering, surgery, right, would have a number of distinct kind of phases.
Right.
Does it help to kind of create this table of contents?
Yes, it does. Yeah, this was.
Wondering if we could combine.
Right. Yeah, because typical. Combinations of tools indicate a particular phase of the surgery. So that was another problem that we wanted to discuss. Then there was also the problem of jitter. We have done the first step in surgery analysis, so which is classification of tools. Then the second one was of tool segmentation. So given a frame, are you able to say that this pixel belongs to tool A and this pixel belongs to tool B and so on? So this helps in differentiating between tool and tissue, and it has extensions towards robotic surgeries as well. So for this, we trained a semantic segmentation network. We tried fine-tuning it to the neurosurgical tools dataset, but it hasn't resulted in much improvement in accuracy. We suspect it's because NIMHANS dataset has much more blur when compared to this, which was pretty much cleaned, and that's why we thought it's a better idea to directly fine-tune to the NIMHANS dataset. So we were kind of skeptical about this. We were not sure how well this will fine-tune without accounting for domain shifts like we had done for classification, but it worked out pretty fine, and we were able to successfully segment tools from background to a good amount, of accuracy. We employed some amount of we employed class weighted loss. So basically, the amount of pixels that belong to a particular tool is much more less when compared to the number of pixels that belong to the tissue. So you give the loss of the tool much more weightage when compared to the loss of the tissue during back propagation. So these were some of the sample results that we obtained. There are some spurious, inferences from the classifier, but pretty much you're able to identify which part constitutes to the tool. And I feel that using an ensemble of methods, we will be able to make a robust enough network for this. So we got an IOU of 0.65 for tool and 0.93 for the tissue. So the inference that we got from all these experiments was this is the first step towards the automatic interpretation of surgical procedures, and this can be successfully used for giving some feedback to the doctors with respect to their surgeries, so that they can correct their procedures or even improvise on their procedures. And machine learning is a very promising tool, and we can effectively use it for this purpose. There are a number of things that this can be extended to. For example, tool tracking, so they don't miss any tools in the body. Blood loss estimation, and it's a very we were even considering another thing of predicting whether blood loss might happen in the future, looking at the video. So this is real time, but it's a question that was proposed by neurosurgeon at NIMHANS, and also surgeon hand jitter assessment. So eventually, in the beginning, the surgeons might have amount of jitter while performing surgery, but through time they might improvise. So if we are able to give we're able to quantitatively measure all these things in a surgery, a surgeon can improve upon these.
I want to thank the wonderful ambience we have here that encourages academic freedom. All our generous cooperative collaborators, our lovely students, our very, very supportive colleagues, and the concept of Samvaad as a forum to present. This is the first time in my 7 years here that I'm telling you about my work. Thanks.